Hi, my name's Marjais Mcbride from the Staley Lab. My year up is on the quorum quenching enzyme SsoPox and its effect on quorum sensing within DSS-induced colitis in mice. Some background on the topic is quorum sensing is a process of communication that is used by bacteria. It involves the release of signals, often in the form of AHLs, which are N-acyl homoserine lactones. These signals congregate with the bacteria they are released from and cause a response by the bacterial group, not just a single cell. This response could possibly be to an environmental change. Quorum quenching is the process where enzymes in the same area of the bacteria have the function of degrading the AHLs. Um, the degradation of these signals causes a decrease in quorum sensing function and could cause a decrease in infections that utilize quorum sensing to spread. Some more background is SsoPox is a quorum quenching enzyme that was used in this experiment. Ideally, this enzyme would have an effect on the quorum sensing processes of bacteria within the DSS-induced colitis mice. DSS is dextran sodium sulfate. A 1.5% solution of DSS was given to the mice over the final 3 weeks of this experiment. The treatment of DSS causes DSS-induced colitis in mice. DSS-induced colitis is a beneficial model for this because of the similarities that it has to ulcerative colitis in humans. These similarities include inflammation of the intestines and damage to the large intestine. The methods in this experiment is the mice were separated by sex and treatment group. So for the male mice, there were two groups of SSO-Pox and drinking water, and then females were one group SSO-Pox, one group drinking water, for a total of 4 groups with 10 mice in each group. For the first 3 weeks of the experiment, the mice were treated with either 2 milligram per milliliter SSO-Pox or drinking water. The food and fluid intake, along with their weight, was monitored throughout the entire course of the experiment, weeks 0 through 6. And then for weeks 4 through 6, all groups were given a 1.5% solution of DSS, along with continued treatment of their SSO-Pox or drinking water. After the 6 weeks were completed, the mice were euthanized and necropsied. The results that we have for the males, or that I have for the males, is I'll start with a principal coordinate analysis, or PCOA, on the left side, which shows the difference between microbial communities. Each dot on the graph represents a sample or microbial community, and the distance between the points shows how related the communities are. If they are farther away, then there is less relation between the microbial communities of the two samples. An Anderson statistical test was run to see the significance between groups on the PCOA, and significance was found between the drinking water mice at different time points, such as baseline and final, along with week 3 and final. This was the same for SSO-Pox, but there was also a significant difference between baseline and week 3. There were not any significant differences at baseline, and then between the two SSO-Pox and drinking water groups, and then at any other time points, there was not significant difference between SSO-Pox and drinking water. The arrows on the graph show the main driver at the starting stages of the experiment before much treatment had been administered is lactobacillus, and then the main driver towards the end of the experiment after the DSS had been added and had been in the mice system for a while was Akkermansia. Looking at the stacked bar chart on the right side, it compares the relative abundance of the 10 most abundant families over each week of the experiment. We can see that the relative abundances of families change as the experiment progresses, with some genera, such as Akkermansia, increasing as the experiment progresses and more DSS is added. And then other genera, such as Lactobacillus, appear to be decreasing as the DSS is added to the mice and the experiment progresses. The results for the females, we'll start again with the PCOA on the left side, where the Anderson test revealed significance was found between the same treatment groups, whether that was drinking water or SSO-Pox, through different time points in the experiment, such as week 0 to 6 and week 3 to 6. This was the same for the SSO-Pox mice within their respective treatment groups. There was also significance at baseline in the female mice between drinking water and SSO-Pox mice, but there was no other time point where there was a significant difference between the SSO-Pox group and the drinking water group. The main drivers towards the latter parts of the experiment after the DSS has been added appear to be Akkermansia and Lactobacillus. The main drivers at the beginning seem to be lactobacillus. Looking at the stacked bar chart on the right side, there are some similar trends to the male group. For females, it appears lactobacillus changes with the DSS treatment after week 3, and also Akkermansia seems to increase in the females like it did in the male group. Lactobacillus decreasing after week 3. For conclusions, there was not significant changes between the SSOPOX treated mice and the drinking water treated mice in either the males or the females. More experimentation could be completed in the future to see if there is a better time frame for SSOPOX, and in order to see significant invisible changes caused by the SSOPOX treatment. And these are my references.